Greetings, biology students. My name is Mr. Sanger, and I'm joining the biology team. So welcome to this, my very first screencast. We're going to be starting a new unit called meiosis. And in the next couple screencasts, we're going to be focusing on these terms haploid and diploid. And we're going to figure out what is the difference between a haploid cell and a diploid cell. Why do we care? And then we're going to be relating those terms to gametes and somatic cells. Now, before we do that, though, I think it would be useful to review a little bit about chromosomes, which you learned in your last unit. So, what I want to do is remind you about the anatomy of chromosomes. And you studied this in the context of mitosis, this idea of cell division where one cell eventually splits to become two. And as you know, the important thing in mitosis is that each of these daughter cells gets a complete set of DNA, because the DNA is the instructions for the cell, and so each of these cells needs to make sure it gets a complete set of instructions. So some terminology here. Things get confusing. There's chromatin, chromosomes. What is all that again? Well, reminder that essentially when we're talking about DNA or chromatin or chromosome, really we're essentially getting at the same thing. All of these things are essentially DNA. It's just a matter of how coiled up that DNA is. So if we take this DNA molecule and we start to wrap it up around the proteins and coil it in this tightly organized way, eventually we get this thing called chromatin. And if we continue to coil it up even tighter, we get this really tight coil called a chromosome. So if the DNA is loosely coiled up, we call it chromatin. When it's tightly coiled, we call it a chromosome. Whichever term we're using, though, we're essentially talking about the DNA, that genetic information for the cell. So DNA replication, you may remember, happens during interphase, that first part of the cell cycle here, when the DNA is stored as chromatin. And since chromatin is kind of loosely coiled up, then we kind of use these squiggly lines to represent the DNA there. And the reason why DNA is copied in this phase is because if the DNA is coiled up too tightly, then it's hard to get in there and make copies of it. So keeping it nice and loose allows DNA replication to happen. And so an analogy for chromatin would be just a bowl of spaghetti. If DNA is represented by a spaghetti noodle, the chromatin would be just this big bowl, kind of this loose coil of tangle spaghetti. And that actually kind of looks like spaghetti, doesn't it? Yum. Now, during cell division, or mitosis, that chromatin is going to coil up into a chromosome. And the reason why the cell does this is because it makes it easier to sort of divvy up that DNA equally and precisely. That chromatin, that bowl of spaghetti, is just too messy to do that. So we coil that DNA up a little tighter into a chromosome, and that makes it easier to divide things out. Now, you may notice that there's little X's here, and then there's little lines here, and that's this whole other level of complexity. So, here's the analogy of the chromatin coiling into chromosomes. So why is it an X sometimes? Why is it a line? Well, remember, before dividing, that DNA is copied. And that, again, ensures that each daughter cell gets a full copy of the DNA. So when it's an X before mitosis, we call this a single chromosome, but there's actually two identical copies of that DNA. So like this half would be one identical copy, this half would be the other, and so we call this an X. It's a replicated chromosome. After mitosis, though, each of these cells here, it's a line. And so again, we still call it a single chromosome, but now it's unreplicated. It only has one copy of that DNA information. And so this always gets confusing for students. If this is an X and this is a line, why do we call them each one chromosome? And a little trick is to count the centromere. That's this little black circle in the middle. There's one centromere here, there's one centromere here, so one chromosome, but because this is an X, it's replicated. Because this is a line, it's not. The term for those two identical copies of DNA are sister chromatids. So each half of the X, essentially, is a sister chromatid, and again, they're identical copies as a result of DNA replication. And then just a reminder that during this part of the cell cycle, anaphase, those two sister chromatids are pulled apart. Very tragic, tearing apart the family. But the sisters go their separate ways, and then eventually, by the end, the cell splits and each daughter cell gets a full and complete set. One final term that can be confusing is this term homologous chromosomes. And so an interesting thing, if you take all the chromosomes in a human cell and you line them up, what you find is that they kind of are arranged in pairs. There's sort of two that are the same size that we call chromosome 1, two roughly the same size we call chromosome 2, all the way on through. And so we call these pairs homologous pairs of chromosomes. Here's a picture of the dog's chromosomes, and you can see in the dog as well, they're arranged in pairs. Those pairs are there because you got one copy of each chromosome from your mother, biological mother, and one from your biological father. And so you get two copies of each chromosome, one from mom, one from dad. These homologous chromosomes, as they're called, the dark purple, light purple, they're the same size, they contain the same genes, but the genes may or may not be identical. So this one may code for, say, brown eyes, and this one might code for blue eyes. In the later unit, we're going to figure out, well, if you get a brown and a blue, how do you decide what color your eyes are? So these are non-identical copies, so not the same as sister chromatids. So I'd like to do, and then the number of pairs will vary from species to species. So what I want to do now is I want to turn to the dot cam and give you a little analogy that I think will be helpful for this. Welcome to the dot cam, ladies and gentlemen. So what I have here are 4 candy wrappers here, and these are going to represent 4 different chromosomes. So we have 1, 2, 3, 4 chromosomes, each represented by a candy bar. Now, if this cell were to begin to divide, each of these chromosomes would replicate. And so when they replicate, basically the DNA, that information is copied, and you would have a situation like this, where now each, I don't know if you can see the whole thing, but now we have replicated chromosomes. We would call the 2 identical copies of DNA sister chromatids. And eventually during mitosis, these sister chromatids are going to separate out from one another. Okay? Now you'll notice that I have a red skittle and a purple skittle. These have the same type of information, but they're not identical. One has the classic flavors, one has the wild berry flavors. And so we would call these homologous chromosomes. Same type of information, they're both skittles, but they're not identical. Same with the Milky Way. These black and yellow Milky Ways are going to be homologous to one another. Same kind of information, Milky Way, but slightly different. Okay? So sister chromatids, identical and attached to one another. Homologous chromosomes, similar information but not identical, not attached to one another. All right, thanks.